Hi. In this second part video, we'll talk about how we look at cells in terms of their size, different kinds of microscopes, and a few things that all cells have in common and what animal cells, plant cells, and bacterial cells look like under the microscope. So for size considerations, let's start big and then get smaller and smaller. So each of these squares that I'm going to show you, the square next to it is 1/10 the size. So we've moved the decimal over 1. So this first square here is 20 millimeters. So the next one is 2 millimeters, and we can see some tissues here, but we still can't see any cells. That's still too big for a human cell. If we go down another tenth to 0.2 millimeters, that would be 200 micrometers. Now we can see cells. A typical human cell is going to be 5 to 50 micrometers in size. So we're right in the range there with our field of view being 200 micrometers in size to see some individual cells. If we go down by another tenth to 20 micrometers, this is about the size of a typical human cell. So you can see that the one cell depicted here is about almost the span of the field of view here. And now at this magnification, we can start to see some structures inside the cells. If we go down by another tenth to 2 micrometers, now we're at the scale that most bacteria are in terms of size. Most bacteria are 1 to 2 micrometers. Some photosynthetic bacteria are a bit larger than that, maybe 4 or 5 micrometers. And there are the smallest human cells are on that scale of size as well. But what we see here in this image, this is not a human cell, but a mitochondria, which is inside the cell. And we're going to spend a lot more time talking about mitochondria when we get to respiration. Now, if you go down another tenth, now it's 0.2 micrometers. That's 200 nanometers. So that's really tiny. And light microscopes can't see anything that's this small. This is beyond the limit of a light microscope, what a light microscope can see. And these little cupcake looking things here, those are supposed to be ribosomes. Ribosomes are essentially really large molecules, so they're quite small. As you can see, the whole field of view here is about 200 nanometers, so each one of these little cupcake things is about 25 nanometers. That's about the size of a ribosome. So that's about the limit. And if we're going to look at things in this size range, look at individual molecules, whether it's DNA molecules, individual protein molecules, individual ribosomes, we need an electron microscope for that. So one question that scientists had started to wonder immediately after cells were discovered a few hundred years ago. So microscopes were invented a little over 400 years ago. That's when cells were discovered. And it's been wondered, why are cells mostly really small? There's a pretty limited size range for all the cells on Earth. They fit into a fairly small size range. Why is that? Why are really large animals made of, or large creatures, whether it's a plant or an animal, made of many cells instead of just one big cell? And the reason is surface to volume ratio. So as a cell gets larger, and think of a cell that's perfectly round. As a cell gets larger, the volume increases much more rapidly than the surface area does. So eventually you reach a point where diffusion across that surface is going to be inadequate to supply the nutrients inside the cell. The limits of the rate of diffusion through a fluid is also a factor. So diffusion through a liquid like water is pretty slow. And if a cell is quite large, it's going to take a really long time for things to diffuse into the center. So generally, the way that cells solve this problem is cells that are really large tend to also not be round. They tend to be long and skinny to increase their surface area. So let's look at the difference here between a cell with a radius of 1, whatever unit that might be, You could think of it as a micrometer, 1 micrometer. And then a cell that has a radius of 10 micrometers. The surface area is going to increase by a factor of about 100. The volume, however, is going to increase by a factor of 1,000. So if the cell is a sphere, that means that it's going to be limited. How much substances can cross that surface per unit volume is going to decrease and decrease. So cells that are large tend not to be round. They tend to be long and skinny or flat as well, like a potato chip. The smallest cell we know today is a type of bacteria called mycoplasma, and it's less than 1 micrometer. It's only 0.3 micrometers in size. And they actually are so small they can't even live independently, and in fact they're parasites of other cells. The largest cells are the yolks of eggs. Reptile and bird eggs, the yolks are actually the egg cell. So all animals have egg cells, plants have egg cells too, and they all tend to be pretty small, except for when they're supplied with a large amount of yolk. And so the largest cell right now is an ostrich yolk. And this is how big it is. This is not a tiny spoon. This is a normal size coffee spoon for comparison. And how does this huge cell get around the problem of diffusion and surface area? Well, it's because the little embryo starts to form and a tiny patch on the top. So there's a tiny little region of cytoplasm on that yolk that behaves as if it's a cell. That's where the nucleus is of the egg. That's where fertilization occurs. That's where the sperm fertilizes it. And that little tiny region of cytoplasm starts to divide and form the embryo. And the embryo gets larger and larger until it engulfs that yolk. And the yolk doesn't perform any cellular functions. Instead, it's just a food supply for the growing embryo. Now, nerve cells tend to be really large cells as well. So you have nerves in your body, neurons, that the cell body where the nucleus is in your spinal cord, but that cell has a long protrusion that goes all the way down to supply muscles very distant from the actual cell body. So the nerves in your legs and in your arms, those individual neurons that compose those nerves, they might be several feet long. So that's quite long. So the longest cell would be a giraffe leg neuron right now. Giraffes aren't the largest animals, but because they have really long legs, they do have the longest neurons. Because the largest animals alive today, whales, don't have hind limbs, and their forelimbs are really short, so they don't have long neurons. But this cell, a neuron, gets around the problem of diffusion and surface area by having an enormous surface area. So your nerves are bundles of these long protrusions called axons from neurons, and they're part of the cell. So to look at cells, we need help because our eyes can only see the very largest cells, and we can't see any structures in those cells with our naked eye. So if we were having a lab in person, we would be using light microscopes. Light microscopes, there are cheap ones and expensive ones. You can get a cheap one for under $100 that works reasonably well. There are compound microscopes that have 2 lenses, and that's typically, when you think of a microscope, you're usually thinking of a compound scope. So it has 2 lenses, it has one lens close to the eye, and then it has another lens that's close to the specimen. And we can change the lens, we can rotate the lenses that are close to the specimen to change the magnification. A dissecting scope is a very useful kind of a microscope. It has a single lens, and so it doesn't invert the image like a compound microscope does. So this image over here, this is from a compound microscope or a light microscope. It just uses visible light to see the object. Electron microscopes, instead of using light, they use high energy electrons to see the object. And because the wavelength of the electrons is much smaller than visible light, we can see much smaller things. So what limits a light microscope is the smallest wavelength of visible light, which is about 400 nanometers, or about half of a micrometer. That's about the size of those mycoplasmas that I just showed you, the smallest cells. That's about the smallest thing that we can see with a light microscope, because that's the smallest wavelength. If anything is smaller than about 400 nanometers, the light waves go around it, and we can't resolve it. At best, it would look like a blurry blob under the microscope. So electron microscopes, because the wavelength is so much shorter, let us see things at much higher resolution. And there's two basic types, and within each of these types there's more little special types of electron microscopes. Electron microscopes are much less common. Chances are you've never used one because they're very expensive, and large research universities probably only have a couple, that many scientists and labs share. They might have a couple of transmission scopes and a couple of scanning scopes. So what does that mean? So a transmission scope, transmission means that you have your specimen is sliced very, very thin, and the beam of electrons goes through the specimen. It transmits through the specimen. That's why we call it transmission. And then there's a detector on the other side, and the deflection of the electron beam gives you your image. A scanning electron microscope, you take your specimen, whatever it is, and you coat it with metal. And then of course it's fixed, or like preserved first. And then you bounce the electrons off of it, and they hit the detector, and that gives us a really nice three-dimensional image of our specimen. And so this creature here, this image was made with an electron microscope. Now, electron microscopes have limits. Because the beam has only a single wavelength, there are no colors. So what makes the colors in visible light is the different wavelengths. So violet and blue light are the shortest wavelengths of light, higher energy waves of light. Once you get into the oranges and reds, that's longer wavelength and lower energy. So that's why you get different colors with a light microscope. With an electron microscope, we have one wavelength, which means that all the images that come out are black and white. So if you want to have an image that has color, you have to add it later. And so in a scientific publication, for example, where it's not allowed to doctor your images with color, you won't see anything except black and white scanning electron microscope images. But on the cover, you might see a colorized electron microscope image. So this is a colorized electron microscope image. And if you're not sure what kind of creature it is, it's a jumping spider. And you can see that it has 8 eyes, and we can see its nice mouth right here. So that's how we know. And jumping spiders are very cute. It's true. So here's all of the wavelengths, or the sizes of things compared to the wavelength. So here's what we can see with a light microscope, and this is the smallest things that we can see are related to the smallest wavelength of visible light. So with our naked eye, we can see things down to about 100 micrometers, or 0.1 millimeters. So that's about the size of the smallest speck of dust that you can imagine. So if you've ever seen dust floating in the sunlight coming in a window, those little specks of dust, they're about 100 micrometers in size. That's about the smallest thing that we can resolve with our naked eye, if you're young enough to not need reading glasses yet. As you get older, you lose the ability to resolve those really tiny things without glasses. So a light microscope will allow us to see things all the way down to about 400 nanometers in size, which will let us see all the cells that exist today. It is difficult to see things at the small end of that spectrum, though. So small bacteria are harder to see with a light microscope, and you need better technique because they're at the edge of what we can see. Most viruses are too small to see with a light microscope. There are some very large viruses that are big enough to see with a light microscope that are larger than 1 micrometer. But the vast majority of viruses are too small and have to be looked at with an electron microscope. So an electron microscope, we can see very small things down to the wavelength of the beam of electrons. And generally in biology, we're looking at individual proteins, individual macromolecules like a DNA molecule, which would be about this size. That would be about the smallest thing that we were interested in. So I will show you some images of very similar organisms that are with different microscopes so you can see what kind of different images we get. So this is a protist. This is a ciliate that is, this image was, the photograph was taken with a light microscope. And you can see that some things are visible. We can see these nice green inclusions in here. It actually has a second kind of protist living in its cytoplasm that's photosynthetic. And we can just barely see the little cilia here around it. And the cilia are actually skinnier than 400 nanometers. So we can see them mostly by refraction of light around them. But we can't make out any detail of the cilia at all. And you can't see much internal structure, but the specimen is alive when the photograph was taken. And that's a big advantage of a light microscope, is your specimen can be alive. With an electron microscope, because it's so high energy, your specimen has to be dead. So here's another way we can use the colors in a light microscope to give us information. We can use something called fluorescent tagging. We can take a fluorescent dye and stick it to an antibody that will bind to a specific protein, and then we can mix that with our cells and light up proteins that are too small to see with a light microscope. So this is really useful. These are lighting up here some proteins in this cell that are, with a light microscope, are invisible. But we can see them if we can change them fluorescent using this fluorescent tagging technique. So this is the same kind of a ciliate, only now we're only seeing these lit up fluorescent proteins. And this kind of image with the black background, it's called dark field microscopy, and we use it for looking at fluorescence. If we take the same kind of organism and we use a transmission electron microscope, so we have to fix it and slice it really thin and then shine an electron beam through it. And this is false color because, of course, electron microscope images are all black and white, but for textbooks, they often colorize them so that you can see what they want you to see more easily. We can see the nucleus is up here. We have some nice mitochondria here in the cell, and you can see the little green circles around the edge here, our little photosynthetic, other species of photosynthetic organism in here. So with a transmission electron microscope, we can see more detail than with any other kind of microscope. Transmission electron microscope, and I will show you over the course of the semester many images from a transmission electron microscope. This is where we get the finest detail, the greatest resolution inside cells. And these kind of microscopes have only been around for about 90 years. So there was many decades of people just looking at every kind of cell they could get their hands on, identifying new structures that were never visible before because we only had light microscopes for several hundred years. So the transmission electron microscope, and also to an extent the scanning electron microscope, allowed the identification of structures that previously were not even visible and were completely unknown. So here's two transmission electron microscope images of cilia. So cilia are, we're going to talk more about cilia later, but I used it as an example in the first chapter for ways we can study evolution using things that are alive today. So here is a cross-section of a couple of flagella, but they could be cilia. They look exactly the same under the electron microscope. They have the same structure. And we can see the little tubes here, microtubules in this 9 doublets and 2 individuals in the center. This is the structure that we see for every eukaryotic flagella and cilia. And then here's some cilia that were sliced a little bit differently. So remember, for TEM, always they're sliced very, very thin. So we're not going to see 3-dimensional structures. So that's why this looks kind of weird. Some of the cilia are cut in cross-section, like these over here, and some of the cilia have been cut longwise or longitudinally. So we can see much finer detail. I mean, the cilia that I showed you in that first image of the paramecium, where you can just barely see them. You can just barely see them. This kind of structure can only be seen with an electron microscope, not with a light microscope. With a scanning electron microscope, it lets us see the surface detail very well on structures. So this is a very tiny animal. It's a human commensal organism called Demodex Folliculararum. It lives in the hair follicles on your face. There are many species of Demodex, and some of them cause diseases, like there's a human disease, scabies, that's caused by a different species of Demodex. Dog and cat mange is also caused by a different species of Demodex. But a commensal organism is one that's not a parasite. It doesn't harm you. It benefits by living on us, kind of like squirrels living in a tree. The squirrel benefits enormously from living in the tree, but the tree, it doesn't really benefit. It's not really harmed. So these are our commensals. And they were discovered that long ago, a couple of decades ago, that they are in fact on everybody's face. They especially are abundant on your forehead and in your eyelash follicles for some reason. And it doesn't matter. You can't wash them away. It's not a matter of cleanliness. This is just a species that has evolved with us, and they live on your oil secretions and dead skin cells. That's what they survive on, and they're so cute. Some other images here for the scanning electron microscope. So here is cilia, which we just saw in cross-section, in longitudinal section with a transmission scope. With a scanning scope, because we don't slice anything up with a scanning scope, we just take whatever we're going to look at, we coat it with metal, and bounce electrons off of it, so we get the 3-dimensional shape. So now, this is one cell. This is the cilia on one single cell, and now we can see their structure in 3 dimensions. This is a marine worm that lives around hydrothermal vents. It's extremely tiny. This whole image is about 500 micrometers in diameter, so it's made of many cells, but that's pretty small. You could see it with your naked eye, but it would look like the tiniest little moving thing you could imagine. That's about what it looks like, 0.5 millimeter in size. So we can see some incredible detail on here, which we could see if we sliced it up and looked at it with a transmission scope, but then we'd have to have a computer to assemble all the images into a 3-dimensional image. The scanning scope gives us really nice 3-dimensional images of things immediately. So if we're thinking about prokaryotic versus eukaryotic cells, remember prokaryotes are bacteria and archaea. Eukaryotes are everything else. Plants, fungi, animals, protists, algae, all those things are eukaryotic cells. We're eukaryotic. So basic features of all cells. So there are some things that all cells have in common, which you would expect, as we talked about. All cells that are alive today can all be traced back to some first cells, those Leuca cells. So these are characteristics also that those Leuca cells had, and all cells have these characteristics because they inherited them from those first cells. So all cells have a plasma membrane on the outside that's made out of a phospholipid bilayer, which we'll talk more about in the membranes section. All cells have a watery substance inside, which we call cytosol. And if you include all the large chunky things in there, then it's the cytoplasm. So cytoplasm is the cytosol plus the ribosomes, the vesicles, the big lumpy proteins, all of that stuff that's in there. So the cytosol is water, dissolved salts, dissolved other larger molecules like dissolved carbohydrates and nucleic acids. That's the cytosol. All cells have at least one chromosome. For prokaryotes, that chromosome is a circular piece of DNA. For eukaryotes, it's linear pieces of DNA. And all cells have ribosomes, and all cells make proteins pretty much the same way. The ribosomes are different enough, though, and remember, this was the basis for Carl Woese's distinction, identifying Archaea as being different, was he was looking at ribosomes. So if I gave you some ribosomes in the test tube, you could tell whether they came from a eukaryote, an Archaea, or a prokaryote. So they work the same way. So it's like having three slightly different machines. They all work the same way, but they're different enough that we can tell where they came from. For bacteria and Archaea, they have no nucleus, and we'll talk in the next video about exactly what is a nucleus in a eukaryote, but it has a membrane around it, basically. And so eukaryotes have a membrane around their nucleus, and prokaryotes do not. Prokaryotes have no internal membranes, mostly. They have their plasma membrane on the outside and no other membranes. Mostly. Like everything in biology, any general statement that you try to make in biology, there's always an exception somewhere. And that's also true for this one. No internal membranes. Well, there's cyanobacteria have some internal membranes. There's a few Archaea that have some internal membranes, but mostly that's the case. They all have a cell wall, mostly, and there's the different types of cell walls is one of the ways that we classify them. They all have a single circular chromosome, they all have ribosomes, and they might have flagella. Most prokaryotes are immotile. They can't swim or crawl. They are just moving around in whatever liquid they're in, floating around, and they can stick to surfaces or not stick, but they can't swim or move on their own. So some have flagella, they can swim. Some are spiral shaped, they can swim. Pretty much the rest of them are immotile. Immotile means they can't move. There's also some confusion often about this central region in prokaryotes, which in the textbook is identified as the nucleoid region. It is not a nucleus. It is the region of the cell where we find the chromosome, the chromosome can move around because before cell division, once the chromosome is replicated, it will move to either end of the cell. Oid, when you see this suffix on something, OID, it means it's like that. So nucleoid just means nucleus-like, but it doesn't actually have a membrane around it. It's just, there's just continuous cytoplasm in here. Here are some examples of different shapes of prokaryotes. Hold on. This is an Archaea, Methanococcus, and it's spherical. And there are many, many species of bacteria that are spherical. And if they are, they may have coccus or cocci, C-O-C-C-I, as part of their name, and that means sphere. So if they are a sphere, they're generally small as well. They're generally 1 or 2 micrometers in size. And we can't see any discernible structures inside of here, no membrane-bound structures. Ones that are bacillus-shaped, that means rod-shaped, and this is another really common shape for bacteria. If it has bacillus in the name, like lactobacillus, that means that it's rod-shaped. They don't all have bacillus in the name, though. Like, for example, E. coli is a rod-shaped bacteria. It doesn't have bacillus in the name. But sometimes the name can give us a clue about what the bacteria eats, where it lives, or something about its shape. So lactobacillus bacteria, that name tells us that they eat lactose and that they are rod-shaped. Methanococcus tells us that it is round and that it produces methane as a byproduct. Some bacteria are spiral-shaped. These can swim if they are spiral-shaped. The bacteria that causes Lyme disease is a spiral-shaped bacteria, and in fact what causes the bullseye rash that you get when you get the tick bite and you get infected with the Lyme bacteria, the ring is caused by the bacteria swimming out from the wound, and as they spiral their way through your tissue, swimming their way away from the wound, they start out in the skin. They spiral their way through your skin. They cause irritation and inflammation, and that causes the redness. So the red ring gets larger day by day as the bacteria swim their way through your skin. Many bacteria have myco or myces in their names, like the group of bacteria called Streptomyces. That's because when scientists were first naming these, back way before we knew that there were prokaryotes and eukaryotes, they were kind of filamentous. So if you see myco or myces in the name of bacteria, that tells you that they tend to stick together into long strands. And these are all electron microscope images, by the way. These three are from a scanning electron microscope, and they're all colorized. And that's why these kind of look like Skittles or something. I mean, they're really not all different colors like this. This colorization, though, emphasizes the strandedness that these form. So as they divide, they just tend to stick together into these long strands. And that's pretty common as well for different types of bacteria. So every cell has a plasma membrane on the outside, and that plasma membrane is made from phospholipids. So here we have a phospholipid bilayer, and we can see the little gray circles here. Those represent the hydrophilic head group, and then the little yellow strands here, those are the two fatty acid tails that are on a phospholipid. So the outside here is hydrophilic, the inside is hydrophilic, and the hydrophobic region is the center where those fatty acid tails are. And this is a very effective barrier in a watery environment between the inside and the outside. The only way something hydrophilic can cross this membrane is if it has some kind of a channel. So this little purple thing here, that's supposed to depict some kind of a protein channel that's through the membrane that's going to allow something to cross the membrane that normally would not be able to. So fat-soluble things, hydrophobic things, often can cross the membrane without a channel, but things that are hydrophilic, this hydrophobic layer is going to stop them from crossing the membrane. So it's a very effective barrier to the outside. So for eukaryotes, there's two kinds of cells that we're interested in, animal cells and plant cells. And this is a typical animal cell with the structures that we're going to cover. The book has a few more than this, but if they're not on this slide, then you don't have to worry about them. I have eliminated a few of them from the ones that are covered in the book. So we'll talk about each of these individually. We'll talk about the three elements of the cytoskeleton. We'll talk about another related part related to the cytoskeleton, the centrioles, which are involved in cell division. The cytoplasm, again, I mentioned. The cytoplasm is just everything that's not in the nucleus. Mitochondria, we'll talk more about them. The smooth and the rough endoplasmic reticulum, the manufacturing center of the cell. We'll talk more about the nucleus and the different parts of the nucleus, the dark region in the center here, which is the nucleolus, nuclear pores, nuclear envelope. We even have a name for the liquid cytoplasm that's inside the nucleus, the nucleoplasm, but don't worry about that word. Ribosomes, peroxisomes, little membrane-bound structures. So one of the things to note about each of these structures as we go through them in the next video is, do they have a membrane? So some of these have membranes, like the Golgi endoplasmic reticulum nucleus. They're surrounded by a phospholipid bilayer. Some don't have a membrane. Microtubules, intermediate filaments, parts of the cytoskeleton, the centrioles, ribosomes, they do not have a membrane. So that's an important distinction between these different cell parts. Which ones have a membrane? Which ones don't have a membrane? Where are they in the cell? And are they in animal cells, plant cells, or both? So here's a typical animal cell. We can see the nucleus here, and again, this is a colorized transmission electron microscope image. And we can see there's a few little structures in here that are visible. Nucleus and nucleolus, and we can see there's a polarity to this cell. There's an inside edge and an outside edge. And here's a nice image of a human white blood cell, and this is a good size comparison because these little balls out here that are surrounding it, those are bacteria. So it kind of gives you an impression of the size difference between a human cell and a bacterial cell. And we can see lots of membranes. We can see the nuclear membrane and all these little stripy structures here, the endoplasmic reticulum, are quite visible in this image, which is, this is again a transmission electron microscope image that's been colorized. And for the plant cells, I took everything off of the diagram that is also in an animal cell. So there's a lot of structures in common between plant cells and animal cells, and also if we include fungi and other eukaryotes, those would pretty much be all the same in all the eukaryotes. But plants have a few extra things. Plants have chloroplasts, which we'll talk more about in the next video. Plants have a large central vacuole, which is usually filled with water, but it can have other things in it too. Plant cells have a cell wall as well. They all have a cell wall. And remember, we already talked about that in biochemistry, that one of the structural carbohydrates is cellulose. That's what plant cell walls are mostly made out of, is cellulose. Polysaccharide, that's 100% glucose, but depending on the species, might not be, if they eat the plant, they might not be able to digest it at all, like we can't digest it. So here's a typical plant cell. We can see, and again, colorized TEM image. We can see little chloroplasts in here, nice nucleus with the nucleolus, and the little orange bodies, those are mitochondria. They're smaller than chloroplasts in this particular image. But you can see there's a lot of similarities between, other than the chloroplasts, there's a lot of similarities between plant cells and animal cells. Plant cells under the microscope tend to look a lot more brick-like because of their cell walls. They tend to be much more rectangular than, not all of them, but a lot of the plant cells tend to look very rectangular under the microscope because of the cell walls that they have. So here's a protist that's photosynthetic. This one is a green algae, actually, Chlamydomonas. If we were looking under the microscope in class, we would have some of these to look at. The flagella are actually very long. They've drawn them really short here, cut off the image. Again, this is a colorized TEM image. But we can see similarities to plant cells, even though this is a single-celled organism that swims around. Again, very similar. It has a vacuole, it has mitochondria, it has chloroplasts, it has a cell wall. It's pretty small. You can see our scale bar there. That's 1 micrometer. So this whole thing is about 8 micrometers in size. So that's pretty small. So in the next video, we'll talk in detail about each of those structures in a eukaryotic cell. The nucleus and the ribosomes and the endoplasmic reticulum and all that. And we'll also talk a little bit more about the evolution of eukaryotes and the first appearance of eukaryotes, because remember that early history of life, the first over 1 billion years of life on Earth, there was only prokaryotes, and then suddenly eukaryotes appear in the fossil record. So we'll talk about what scientists think happened for the origin of eukaryotes.